Hello everyone, welcome back to one more video session. In this session, I'm going to talk about very important concept, that is the structure of polynucleotide chain of DNA and RNA, because both are same. When you just take the structure of the DNA, this is the structure of the DNA, where you could see two strands. This is the one strand and this is the other strand. What is this strand? This strand is nothing but polynucleotide chain. Polynucleotide chain. There are so many basic units of the polynucleotide chain. We call them as nucleotide. Many nucleotides combine together to form a long chain, and that chain is called as the polynucleotide chain. If we just concentrate on only one particular unit of the polynucleotide chain, let me just write. You may get this question for 1 mark. Name the basic unit of polynucleotide chain. The answer is here itself. That is the nucleotide. Done. Okay, no need of thinking. Poly means many. Nucleotide, many nucleotides finally makes polynucleotide. And now there are three components. The polynucleotide chain, particularly I am talking about the nucleotide, has three components. One is nitrogenous bases. I will just write in short NB, nitrogenous bases. The second one is a pentose sugar, I write BS. The third one is a phosphate group. Now nitrogenous bases, there are two types of nitrogenous bases, that is purines and pyrimides. You know about it. Even I have discussed about it in my previous session also, pyrimides. Purines are of two types, that is adenine and guanine. These two nitrogenous bases, that is the purines, are normally seen in the DNA and RNA, which are common. Whereas pyrimides, that is cytosine, thymine, uracil, these three things, particularly in DNA you see cytosine and thymine as pyrimids. In RNA, instead of thymine, uracil is there, that is cytosine and uracil will be seen. Here I just want to give you one more information. It could be asked for your part A section, that is for 1 mark. What is the another name of thymine? Thymine is another name. I just write thymine is also called as 5' methyl uracil. The another name of it. Hope you can see this 5' methyl uracil. Just remember, it could be asked. What is the another name of thymine? There is no relation between this thymine and uracil. Both are structurally and chemically different. Fine. Okay, so this is the thing. Now, we'll talk about this pentose sugar. You know about the pentose sugar. I just draw a pentose sugar here. This is better. Okay, so this is the pentose sugar. This is the first carbon atom. This is the second carbon, third carbon, fourth carbon. And here you see the fifth carbon. We just write C here. Whenever the pentose sugar comes, obviously, when you look into the nucleotide, one pentose sugar should be there. And here to the first carbon atom, you see a nitrogenous base. It could be adenine, or it could be guanine, it could be cytosine or thymine. In for RNA, it could be uracil. Is now attached here. That means a nitrogenous base, it could be adenine, guanine, cytosine, thymine, whatever it is. I just write NB. Or maybe like for our understanding, simply write A for adenine. So this adenine is now attached to the 1C. That is the first carbon of the pentose sugar. This is very important. Remember it. Now, if I talk about the purines, I just write this first carbon atom is attached to the 9th nitrogen atom of the purine. Okay, we write. If I talk about purine, so 9N to 1C of pentose sugar. This is the 9N of the purine. That's it. So this is the linkage. If I talk about adenine and guanine, that is purine. If I talk about the pyrimides, I just said P here. Okay, so that is the first nitrogen atom which is linked to the first carbon atom. That means one thing is sure, that whatever the type of nitrogenous base, it is always attached to the first carbon atom of the pentose sugar. This is common. Whereas, and if I talk about this one, nitrogenous base, purines or pyrimides, for purine it is 9N, for pyrimides it is the first nitrogen atom, that is the linkage. And these two are linked by this linkage is by a type of N-glycosidic linkage. Very, very important. N-glycosidic linkage. Very important. Please remember this, very important, N-glycosidic linkage. So a nitrogenous base is now attached to a pentose sugar by N-glycosidic linkage, and now these two structures now are called as, for example, I write here pentose sugar plus any type of nitrogenous base through N glycosidic linkage is now called as nucleoside. Very, very important. So you get a question, what is a nucleoside? When a pentose sugar attaches to a nitrogenous base through N glycosidic linkage, it is called as nucleoside. You have to mention that linkage or bond. Okay, so that's it. That is nucleoside. So we know about the pentose sugar. If it is DNA, obviously we know that oxygen is present in the second carbon atom position, whereas oxygen is absent in DNA. Whereas in RNA, oxygen is present. That is hydroxyl group is there in the second carbon position. That's it. That's it. We know about. That's the basic. Now let us talk about this. What is this? The phosphate group. This is the phosphate group. Always remember that the phosphate group is attached to the fifth carbon atom of the pentose sugar. So I just write P here. So here, let us imagine or assume that this is the beginning of the DNA chain. See, you could see a double helix model of the DNA. I am talking about this particular structure. Let us assume that this is the beginning of the DNA. And now this is the end of the DNA. I am talking about single polynucleotide chain. So here what you see is a free phosphate group is in. This is free. That is free. That means this one end of the phosphate group is not attached to another end or another pentose sugar. This side, it is free. So as it is attached to the fifth carbon atom of the pentose sugar, always remember that the DNA starts from 5-2. We don't know. We'll see, but actually answer is 5- to 3-. This is very, very important. Always remember a number that is 53. 53. Remember this. It is applicable for replication, transcription, and everything. That's what. Like you get a question like, what is exactly the direction of transcription? 5- to 3-. What is the direction of replication? 5- to 3-. Or what is the the position of the DNA or DNA strand? That is 5- to 3-. How exactly DNA runs, in which direction, that is 5- to 3-. All these types of questions could be asked. Very simple, this is the basic. Okay, but now here the second phosphate group of the polynucleotide chain is connected to 3 carbon atom of the pentose sugar. So it's going down, we'll see it. So just write, this is the 5th carbon atom of the pentose sugar, and see here what you see. Like this, and here this is the first carbon atom. You could see any type of nitrogenous bases. It could be guanine, cytosine, whatever it is. So it runs. This is the third C, and again what you see, another phosphate group. That means it is understood that one phosphate group is attached to the 5th carbon atom of the pentose sugar, whereas another phosphate group is attached to the 3 carbon atom of the pentose sugar, whereas this phosphate group, another side of it, is attached to the 5th carbon atom. So that means the linkage. Let us talk about the linkage. So nucleoside, this is nucleoside. I am talking about this and this. Nucleoside is now connected to this, a phosphate group here, one phosphate group, through phosphodiester linkage. Shall I write here? Okay, we write here phosphodiester linkage. Very, very important. Please learn it. A nucleoside is attached to a phosphate group by a phosphodiester linkage. So now these 3 components, a phosphate group, a pentose sugar, and a nitrogenous base, now forms the basic unit of the polynucleotide chain called as nucleotide. Now many nucleotides, see, this is the second nucleotide, correct? This is the second nucleotide. Now 2 nucleotides are now attached by the same phosphodiester linkage, but whenever you get a question like the 2 nucleotides are now attached to one another by what type of linkage? Don't write only phosphodiester linkage, you have to write its direction too, that is 5- to 3- phosphodiester linkage, that is between 2 nucleotides. That's it, simple, very easy, right? That's it. Why you have to mention 5- to 3-? Because see, one phosphate is attached to the 5-, whereas the other phosphate is attached to the 3-. That's it. So it goes like that. See, again 5-, 3-, 5-, 3-. It goes like that. 5-, 3-, 5-, 3-. It continues like that. Now, what is there or what is present at the beginning of the DNA? That is free OH. What is there at the end of the DNA? That is the terminal of the DNA. This is the beginning of the DNA strand, and this is the end of the DNA strand. I am talking about single strand, that is one polynucleotide chain. What do you see? Let us assume that this is the end of the DNA. It goes like that. It's very, very lengthy, but let us assume that here there is a OH group, that is free OH group, at the third carbon position or atom of the pentose sugar. That's it. So a free OH will be seen. Remember. So 5-P, that is a free phosphate group is there during or at the beginning, whereas at the end or terminal of the DNA contains a free hydroxyl group at the third carbon atom of the pentose sugar. So this is all about the polynucleotide chain. This is it. Hope you understood this concept. Very important. I have covered all the basics and even some concepts for meat also. Everything is covered.